Hi everyone, my name is Amin. I'm a graduate student at Columbia University, and I'm going to present my accepted paper in MICCAI titled Probabilistic Joint Segmentation and Labeling of C. elegans neurons. To give you a quick bit of background, C. elegans is a transparent nematode with a simple nervous system consisting of 302 neurons. The nervous system is stereotypical, meaning that same neurons almost always appear in the same location relative to body. And neurons have names, and almost every single animal has the same number of neurons with the same properties. Because of this reason, a recent biological technique could develop a differential coloring scheme using fluorescent proteins for coloring different neurons in a deterministic manner. This enables the unique identification of neurons as well as the development of a statistical atlas that encodes a canonical position and a canonical color for each neuron. An important question given this biological technique is to develop methods for automated identification and segmentation of neurons, which can enable conducting high throughput experiments. So there's a long-standing literature on the segmentation methods, but the problem for C. elegans is a little bit different because of the following reasons. So we're dealing with large data sets, meaning that we have volumetric images in multiple color channels. The images contain background artifacts such as gut cells and lysosomes and background noise. And there's not a lot of training data available for the segmentation. And we also want a method that makes use of the recently developed statistical atlas of neuronal positions. Here I've included a very high-level summary of this existing approaches for the segmentation. Deep learning methods require training data. Watershed methods are not robust to noise and artifacts. They also require a filtering step. Graph-based methods are not efficient. Mixture models, although they're used in the context of segmentation, but they do not make use of a statistical atlas. And finally, in the last row, we have the multi-step approaches that consists of a filtering step to filter non-neuronal objects, and then a segmentation or a detection step that find the candidate locations of the objects or neurons, and the final step is labeling, which is to find the correspondences between the objects in the image and the neurons in the statistical atlas. In the last column, I've included whether these methods are capable of solving the labeling problem at the same time that they solve the segmentation problem or not, and only the last row is capable of doing that. So here's how the data looks like. So in the second row, we have the data. This is a volumetric neuropal image that is max intensity projected onto the Z dimension. In the first row, we have a statistical atlas of neuronal positions and colors, and also the positional covariability, and that is represented in these ellipses. So our goal is to solve the segmentation and labeling problem at the same time. The segmentation problem is to just find the regions in the image corresponding to each neuron, and labeling is to find which neuron that segment is representing. We also want to disentangle the background pixels from the pixels corresponding to the neurons. To solve both of these problems simultaneously, we introduce a statistical model, which is a mixture model, and it has two different parts, which is a prior distribution, and we want to use the statistical atlas in the form of a prior distribution in our statistical model, and the likelihood, which is just a mixture model. In the next slides, I'm going to explain in detail what each of these parts demonstrates. So the first part that I'm going to explain is the prior distribution. So our prior distribution is a normal distribution given the parameters in the atlas. So we have parameters for the neurons, for the neuronal centers. Mu1 to k are three-dimensional vectors, x, y, z, corresponding to each neuron. And then we also have a shape parameter, which is a 3x3 covariance matrix for each neuron. Here, mu a and sigma a are the atlas parameters. The a superscript a denotes the atlas, and mu a1 to k corresponds to the neural centers, the canonical neuronal centers in the atlas, and sigma 1 to k a are the neuronal positional variability. We also have a parameter beta that determines what is the affine transformation between the statistical LS and the current image. Since the LS lives in a canonical space, we need to align it to the current image to solve this problem. Notice that the mu1 to k and sigma 1 to k and beta are the parameters that we're going to infer, and mu a and sigma a are the parameters that are given by the pre-constructed statistical LS. So given this prior, our likelihood is just a mixture model. In our likelihood, we have components that correspond to neurons and components that correspond to background. Each observation is a 6-dimensional vector. Each observation corresponds to a pixel, and the 6-dimensional vector is just a concatenation of the location of that pixel, xi, yi, zi, and the color of that pixel, ri, gi, bi. And we have mixture component weights for both neuron components and background components. Our assumption here is that the neuron pixels are normally distributed both in color and space, while the background components are normally distributed in color and uniformly distributed in space. A common approach for solving this problem is called expectation maximization algorithm. And in order to infer the parameters of this mixture model, a new latent variable z is introduced. This is called the assignment variable, and it's a discrete variable. So if we condition our joint model on the assignment variable, we will get back one of the mixture components, as shown here. Once we introduce this assignment variable, we can compute the complete log likelihood, which is a function of the parameters, and is equal to the expectation of the log joint distribution with respect to the posterior distribution over the latent assignment variable. Given this complete log likelihood function, the E and M steps of the EM algorithm are described as following. E step is to compute a matrix called gamma by evaluating this Q of theta given the previous value of the parameters. This gamma matrix is called the responsibility matrix. It has rows that are equal to the number of the observations or pixels and columns that are equal to the number of the components. And we can think of this gamma matrix as some sort of probabilistic segmentation of the image, because the element I and j of this matrix corresponds to the contribution of pixel I in components j. The M step is to just update the parameters by maximizing the complete log likelihood function. So here's a summary of what I just described. Gamma is called the responsibility matrix. It provides a probabilistic segmentation of the image, and it determines what is the probability of pixel I being assigned to a neuron or a background component. However, we have more information that we want to incorporate in this problem. For example, we know that the components that correspond to background are larger than the components corresponding to neurons. We also know that this gamma matrix has this property that the segmentations are usually diffused in space. So the number of pixels that have non-zero probabilities for each component are fairly large. However, we want to have sharper segmentation with more local components. For this, we developed a new algorithm that we call synchron expectation maximization, which is very similar to the vanilla EM algorithm, except that we have a step here to enforce row and column sum constraints to the gamma responsibility matrix. The M step is exactly the same thing as the M step of the vanilla EM algorithm. To study the performance of our method, we have metrics that correspond to the labeling. So the MSE is how far the infer centers are from the ground truth centers, and the accuracy is just a thresholded version of the MSE, where we just threshold the centers that are closer than a certain value to the ground truth centers, and we call them a hit. And then the accuracy is just the percentage of the hits. To study the segmentation quality, we introduced this new metric called location spread, which determines how spread and diffuse the components are in space. We compare our algorithm against multi-step methods of filtering and then detection and then identification, and also vanilla expectation maximization. We used head and tail images of C. elegans for our comparisons, In our experiments, we used three background components to mimic dark, green, and blue artifacts. Here's an example of how the data looks like. The first panel is the data. The second row is just a zoomed in version. And the third row is a segmentation given by SEM and VEM algorithm. This is another demonstration of the quality of the segmentation. The left column is the SEM, the right column is the VEM, and each row corresponds to the probabilistic map for each component. We also studied the performance of different methods quantitatively and showed that the accuracy and MSE is better for the SEM, both in head and tail compared to other methods. We also looked at the location spread for SEM and VEM and showed that the components given by the SEM are more local in space. Finally, to conclude, automated identification and segmentation of neurons is very important for many biological applications. Current approaches either aren't scalable to large data sets, they require training data, or they have multiple steps, each of which are prone to errors, and they do not make use of pre-constructed atlases. We formulated the joint segmentation and labeling problem as a statistical mixture model, and we developed an algorithm as an alternative to vanilla expectation maximization, and we showed that it provides better accuracy and sharper segmentation. Future directions include better alignment techniques as well as better registration models instead of an affine registration, such as non-affine or piecewise linear methods. I would like to thank all the people who contributed to this work and you for listening.